Mechanism of humoral mediated immunity, otherwise called as the antibody mediated immunity, is the important short notes in the university examination. It may be asked as the long question. The humoral immunity. Humoral means body fluids. The antibody is enter into the body fluids, that is product against the bacteria. So it is called as the humoral immunity. So antibody mediated immunity is the other name of the humoral immunity. So antibody is act against the bacteria or antigen and kills the bacteria. It is produce the antibodies. Antibody is produced from the B lymphocytes. B lymphocytes. The B lymphocyte is converted to the plasma cells. It produces the antibody. Humoral immunity defends against the bacteria, bacterial toxins, and rarely viruses. Development of B lymphocytes. The B word is derived from the pursa of papricus in the bats. So the B lymphocyte also develop from the bone marrow of the fetal liver, bone marrow or beetle liver. It is developed from the pluripotent stem cells and the lymphocyte stem cell. From the lymphocyte stem cell, D cell enter into the thymus, produce the D cell. From the lymphocyte, from the bone marrow itself, the B cell is produced, that is enter into the lymphoid tissue like lymph nodes, spleen, blood and lymph. The bacteria enter into the body, that is phagocytosed by the antigen presenting cell. The antigen is fragmented into the peptides. The peptide fragments are carried on the surface of the antigen presenting cell, that is bind with the beta cell receptors that stimulate the proliferation of the beta cell, that is called as the clonal selections. It produces a more number of the beta cells. The beta cells are converted to the plasma cells and B cells and memory B cells. The B cells are converted to the plasma cells. The plasma cells produce the antibody. That antibody enter into the blood and enter into the circulation. Consequences of antigen-antibody binding. After antibody is bind with the antigen, it produces a clumping, that agglutination. So that lies the antigen and the antibody cover the antigen. So it makes a tasty material. It is engulfed by the macrophages, that is called as the opsonization. And antibody neutralizes the antigen and kills the bacteria. And immunoglobulin, antibody like immunoglobulin IgM activate the complement system that kills the bacteria. And antibody dependent cell mediated cytotoxicity lyses the bacteria. In this slide, consequences of antibody with the antigen binding is explained in the flow chart format. That the neutralization, the antibody neutralizes the viruses and bacteria and agglutinate the bacteria and precipitate the bacteria antigen and activate the complement fixation and lyse the cell. Here the neutralization, agglutination, precipitation is finally engulfed by the macrophages. Summary of the humoral mediated immunity. It is almost like the cell mediated immunity. When the bacteria enter into the human body, it is carried by the antigen presenting cell. It converts the lyse the bacteria and fragment the antigen to the antigenic fragment, peptide fragments, that is carried into the surface of the antigen presenting cell, the MHC2 protein that is attached with the P lymphocytes. P lymphocyte is stimulated and proliferated, produce a more number of the P cells and memory P cells. The P cell is converted to the plasma cells. The plasma cell produce the more amount of the antibodies. Each plasma cells produce more than 2000 molecules of the antibody. That antibody enter into the circulation and lymphatic system that kills the bacteria by the accumulation, precipitation, neutralization through the complement fixation and antibody dependent cytotoxic activity. Thank you for watching this video. If you like this video, share your friends. Those who are not subscribed this channel, please subscribe. Thank you once again.